information from there, but Chelsea will talk about walruses and how she's worked with local hunters in St. Lawrence Island, as well as up at Utqiagvik. So we look at time series, which without, because no one country can afford to have sampling every month of the year. And what this is just looking at when there's a lot of sea ice, there's more ice algae. It's more labile, it means it's more fat, and that's good then for growth, and for a lot of that carbon goes to the bottom. And what we're starting to see is that less of this ice, it will have an impact on the type and quality of food available for some of the either the zooplankton or the animals that live in the water column. And Chelsea will be able to say some more about that. I don't expect you to read all this, but I just wanted to show you what we are doing nationally and internationally. Depending on the time of the year and which country's involved, we go and sample some set sites, and then we compare the results. And we agreed that we would look at temperature and salinity, currents, which are important to know where the material goes to, where animals are following it, or what the temperature and salinity is. And then we're looking at chlorophyll, which is the plant life. We look at who lives in the water column, who lives in the sediments. And then we're working with other marine mammal and seabird surveys. So we work with U.S. Fish and Wildlife Service, and they do seabird surveys, and NOAA puts out for marine mammals on many of these ships. And then periodically is bottom trawling, and that's the NOAA activities. But at the same time, we have a lot of this information coming from gliders, which are autonomous vehicles. There are moorings, which means that there are measurements going year round near the bottom of the ocean. And then the big thing is that we place these in areas where there's a lot of productivity. We can track from the lower to the upper water column. A lot of countries involved in that. And we just had our 5th international meeting down in Seattle to try to share those data sets to track what the status and how these changes are occurring in the Pacific Arctic. So this is from last year. You all know what the surface water warming, when we've been seeing this in the last couple of years. But what was amazing in the 2018 was the bottom water. For those of us who study animals in the water, in the lower part near the sediments, it was 3 degrees Celsius warming, which is about 4 or 5 Fahrenheit difference. And what this is comparing July. It's an anomaly, which means the difference between the average over from 2010 to last year. And you can see all this warm water, which is all being way up by 5, which is about 8 to 9 degrees on the surface water here.
The system has detected that a minimum number of lines are connected to this conference.
Okay.
Do you want to continue your meeting?
Yes.
Press 1 to confirm. If there is no response, the conference will end.
I think you have 1 call or 2. Yeah, I answered yes, Gabe. Just keep doing what you're doing. And then this basically just looking at the 2 years, you can see with this really warm water that we're seeing southwest of St. Lawrence Island, but even all the way up into the Chukchi Sea. So the temperatures are warming, melting, helping melt the sea ice, but having a big impact on the type of the quality of the food for the animals that live here. So this is just one example. This is south of St. Lawrence Island. These are the threatened diving sea ducks, the spectacled eiders. And we had an opportunity for, this is high up in the air with the BBC. And the question was why the birds, why they were scattering like that. And they were looking at the breeding impacts, and it was the walrus coming up. And so this was something that looking at it, the fact that they like in the Antarctic where they're feeding, they can feed on, walruses obviously can, they are known by traditional knowledge to also feed on seals and so forth. So we have 3 dominant species we've been looking at. This is prior to the period we have. This is almost a decade ago, but this is the cold water that is produced in the winter time south of St. Lawrence Island. When ice is formed, salt is rejected, it goes down, it can have really cold water and it doesn't freeze. And all of these animals are adapted to living at those low cold water temperatures. But it's this thermal barrier that kept these demersal fish from the south, which is your Pacific cod and the pollock that you're starting to see up here now, from coming that far north. And so as a result, a lot of the animals that live in the sediments that walruses, gray whales, bearded seals, diving ducks feed on have been able to be really high populations. But as these big these animals that feed on worms and clams, they're competing now potentially with a lot of this benthic feeders. Just an example of what you all see in 2019. This is just the sea ice extent. It went away in February. You know, it came back again in April on average, and this is in the area south of St. Lawrence Island. In 2018, it went away in March and didn't come back. That's when this thermal barrier really broke down. And this is compared to what it had in the past as the mean. And we have this colder winter going on this year, but as the locals were saying too, it's very thin ice. And the question is how much will build up this year and how thick it will be and how quickly it'll go out of the system. And on the right here is what we see from satellites from the chlorophyll. And the big question here is, this is 2019. We had, last year we had a peak in the spring, but then it's really variable. And the thought is that without that sea ice there, we won't necessarily have that really nice spring production of organic matter that's important to feed this, get the clams going. And that in fact, the production may not be as high in certain regions, south, for example, south of St. Lawrence Island, which will have an impact on the type of zooplankton later on in the spring. Just some, I don't want to go into too much detail, but just to point out, we run these standard stations, and the brighter the red color, the warmer it is. And you can see how warm it's already up above 10 degrees Celsius, which is pretty high for surface water. Most of the chlorophyll was down, what we call, it had already settled down, but it's growing. And this is something you don't necessarily include on your satellite observation. So it's important that we know that this is going on there. There's enough light going through because it's in the Arctic areas. That's what we call subsurface chlorophyll. Yes. Well, this is the latitude.
The places where the ship stopped in blue dots.
Yeah, so this is the blue dots. So this is the coming up. Yes, so we have within here, there are 5 stations. We come up on this line. This is the traditional hotspot area for where we've seen these clam populations and also where the diving sea ducks have traditionally have been down in here. And then these ones we move up into this, which is traditionally where the amphipods are, where gray whales are feeding. So that's the spatial location. And this is, you're looking from surface to bottom, and you're looking from south to north for latitude. So when we go in this direction from the southwest to the northeast, this is the line you're seeing. And you're getting that surface water. And this is the oxygen that's produced by the plants that are associated with that fluorescence. When we look at the time series, so this started working on these sites. We picked out 5. It takes in this embedded area that are offshore that we traditionally have all been clams. And what we've seen is that a reduction of the biomass. So this is how much they weigh and a certain volume of sediment we bring up. And these, what it's saying is that most of the hot spot that we've found that 5 stations would record has contracted northward. So that the feeding patch that these birds are going after, or even the walruses, is contracted northward. So there's less down here. These have been moved into worms, which are fine for bottom feeding fish, but they're not what a diving sea duck goes after. And walruses will go after, as you know, bulk of different types of food. But this is one that most of the clams that the bigger clams are up in that northern region. And these are some of the common for the eiders because they have a set length of which they can get down their esophagus, down into their stomachs. So we're seeing this northward movement or northward contraction, not just south of the island, but north of the island, and then also in the southern Chukchi Sea. The only thing to take home from this figure is this decline over time. This is in the late 1900s. These are the time series sites. So these is the average is the black, and this is this contraction. Northward are the brown clams, and the yellows are the worms. The main thing to show you is that this used to be a really broad and higher amount of food for these organisms, for these ducks and for walruses, and it's now a smaller, and the composition has changed. So when we talk about changes, it's not just how much is out there, but it's who is out there, who's in the community, because some of these animals have more lipids in them than others. So a prey, if, for example, if a duck has to dive down 60 meters, 150 feet or something like that, and come back up, if it has less of a amount, picks up a clam that doesn't have as much calories in it, it's not as fat, it's going to spend a lot of energy, but get less from the return. And this is something that we're seeing also with walruses that are coming ashore, that they have to spend a lot of energy to go out and feed on these richer clam beds than they had in the past when they could ride the ice north. So that's one example of what we're seeing in the bottom.
Jackie,
Yeah, sure.
talking about a range shift of a sessile organism, it sounds like a recruitment issue. Is it related to
Well, they do produce their young.
reproduction and growth? And the clams are.
Well, I think the thing is that these clams are actually taking advantage. It's also sediment grain size. So these are finer sediments now. So some of these animals that are taking over, they're subsurface deposit feeders. So I don't think that, the type of sediment that there's available now is something that is more conducive to a certain type of species like the worm versus a certain type of clam. So the recruitment part, is it something that the birds have taken away all the adults? There's a potential possibility for that, but there's not enough birds in that certain area to have consumed everything, unlike certain regions off of Greenland. So to ask whether it's a recruitment type thing, a lot of this has to do, I'm saying this more has to do with the fact that the currents are changing enough and a lot of the settling out of the carbon, even though the carbon is high, some of it's not conducive to what those organisms, and their surface deposits, so they can be smothered, compared to a polychaete that can live deeper in the mud. Okay?
Not that I'm gonna ask questions right now, but so what I'm hearing is that there's less clams, less food on the bottom. Would that also have something to do with the fact that we have all these Pacific cod and pollock and things that have come from the south that eat the bottom?
Yes, that's one of the things. I mean, because what I'm understanding that inside the stomachs of these Pacific cod and pollock are a lot of worms, and so they're bottom feeders. So used to be the cold thermal would keep out those big mouths, and you would just have the walruses, the diving ducks, and so forth. But now you have a pressure. You have those animals, and then you have those fish feeding, and then they're coming in and feeding a lot on these marine worms. So I think that there's a top down, what we call from predators on top, and a bottom up on the hydrography, and both those things are having an influence on this system. Yeah, clams are taken, and these are this is this trawl survey from October 28th, and you can see the contraction of there's a Pacific cod because the thermal barrier is reduced, and the pollock. And we know now that the pollock, and with the long lining that's going on north of the island to Bering Strait, they're there, and they're there until the ice comes back down. So now we have these big mouths in the system where they didn't used to be in the system. And so there are going to be winners and there's going to be losers into this region. And then if you go north of the island, we have two standard lines. What you're seeing here now, you're going from west to east. Warm water again on the surface, but we had a bloom going on. And because the way you get oxygen in the surface water is to have a lot of plants growing. It's just like when you have it in your plants in your house. So it's producing that oxygen. So this was done with there's enough nutrients there. And so that's something that is we were able to see during that one period of time and just on this one transect line. And this is the area that's traditionally where we would have the gray whales beyond having walrus. And what you're looking here is 4 decades of data. And the thing I would point out is the pink. The pink is where the small, what we call amphipods, small shrimp-like creatures that the gray whales would feed upon. And they were spread out more in the 70s. And the boxes are a time series because we can't go back every year and do all of this sampling. But what you're seeing by the 90s, we were already seeing changes before the thermal barrier went away. So something was going on in the ecosystem starting in the 90s. And this area moved from being food for gray whales to more for sculpins. So these type of, they're called amphoretid amphipods, but they're found in sculpins, in the stomachs of sculpins, particularly around this side of St. Lawrence Island. And then when we get into the 2000s, we then moved into this one changing to clams. And this one staying with polychaete marine worms. And now, if you look at this, you can't really see it. Again, we're having this contraction northward of species. And a lot of this has to do fast currents coming in here. We have more settling out in this region here. And the amphipods, these shrimp-like animals, they like to have a lot more sand. And but they need to have fine particles to glue them together with their mucus. And it looks like this area is more conducive for that. And this area is getting change in the speed of the currents and the type of animals that are better competing now versus these particular amphipods, which are in the upper right. And then the gray whale is on the upper left. One thing I would point out is what we found out. I mean, the eiders are usually overwintering here, but some of them with the lack of sea ice started migrating earlier, and they're being found on the Russian side. And they happen, and if you, some of the data we have, there are more of these clam species. So if they're up there, they're feeding. The winds may change, they may build ice again, but if they're finding places they're open water to feed, they're not coming back down to that area, and they're actually spreading, potentially spreading their range out. And this is once again, just main thing to look at the black is these changes that we're seeing, this contraction northward. The pink are these small amphipods that the gray whales go after. And these populations of what we're also seeing, finding sediments because of slowing currents north island are where there's a buildup of these marine worms. So it'll be great for pollock and Pacific cod to come in there, but it'll start changing but there's not as many as you go into the northern area. So there's a competition between fish and some of these really high organisms like for example, the gray whale, and they're spending more time than going northward. So environmental stressors, I mean, one of the things that happens when you bring warm water, I mean, we're seeing acidification. That's when you get more carbon dioxide in the, so it can be corrosive. So it's more what we call acidic. This is just an example of off of Utqiagvik and over up of, we have Hannah Shoal up here, where we have a lot of clams, and they take up oxygen. And if the currents are slowed down, the carbon dioxide can build up. And if with that you can have some corrosion of clams. Now we've seen 2 types of clams up there that are really important for the walruses when they come out to feed in that area. One of them was impacted and their shells got thinner the more time they spend in this acid water. But another clam they were what we call resilient. They had thick enough shells, they're able to survive. So one is going to become more abundant potentially than the other. Another thing that's harmful algal blooms, you probably have heard about it. It's a toxin. It's increasing in this area. We have warming seawater, and they're finding Actually, this was found by the Japanese back in 2013. There's a bed. What that means is that they make when this algal species over the winter, they cyst. They make a little, like a casing, and they get down in the mud and they overwinter there. And then when the waters, if they're warm enough, they come up and they bloom. And that's what's been happening to some of the, been observing that, Don Anderson and some of the folks from NOAA. So this blooming, you hear it coming off of Florida, you hear it in the Atlantic sector and the Gulf, but it is up here. It's a dinoflagellate, and there's different types of toxicity that it has. And so this is something that people are, and there's a whole network involved in, and as sampling, as oceanographers, we're going out and collecting a lot and passing them on to some core people to do the toxicity studies. But it's important because they close down fisheries, you know, in other parts of the US if it reaches a certain level. Yes?
Has those algal blooms been in the mud down for warm water all these years, or it migrated up there and then sunk when the water got cold?
That's a really good question, and there's no answer that I know of on that, whether they, it's probably because if they found them in 2013 in the mud, so they were there before we even got, quote, this warmer water. So the question is, has it been happening and then water wasn't warm enough for them to make a bloom, or nobody was looking for them? And I think, you know, it's a combination of both of those, okay?
So there has been information for marine mammals that were sampled in the 2000s, that this is not new to marine mammals in our throughout the state of Alaska. Saxitoxin and another one, not new to the region. There is some local reference names that indicate Ignara. If you look at the National Park Service book, Ignara just northeast of Shishmaref, about 18 miles. And the place name book says this is a place where several people died after red tide. So we do have some inkling. I don't think anybody's documented it. I think that is probably that definitely needs to be done, but I don't think it's brand new. What is new is that the warming conditions allow for these out of control growth events to happen, and the temperatures are finally getting warm enough to wake up some of the cysts that have lain dormant on the seafloor. If that helps, right?
Right. And so, you know, there's no absolute answer, but the fact, and that's where you need to get some of this back data that people have been talking about or are in the books to look at, that's not, that provides some perspective of this. But I do think that you know, the fact that you have finding these beds, they're just waiting there, but they're only going to come out if the water's warm enough. This was a cruise we did on the Healy last year, back in August, and this is part of the network. We collected in our benthic team clams and worms, and we'll continue to do that. We're one of a network, and then they were run for looking at toxicity levels. And just to give you an idea, so this is the samples from the cruise, 80 micrograms value for shellfish is about 800 nanograms per gram. The point is that this red value is way above anything anybody should eat. And this was found north, here's St. Lawrence Island down here on our study, on one of the time series sites, northwest, and one of the clams. And you can see that the ones that are the lighter yellow, they're below the level that would have an impact on having presence of food web. That would be food web. Huh?
Below the seafood safety level.
Right, below the seafood safety levels. But you can also see them we're picking up as you go through Bering Strait here. And here's Point Hope is up here, right here, Cape Lisburne. And this is what was called from Ledyard Bay. So there's another one there, but pretty close in this hot spot we have in the southeast Chukchi Sea, which is an area where we have, you know, accumulation of bowheads, gray whales, humpbacks now coming in. But what we were looking at and some values up here, but mainly in the clams, except they're, you know, the lighter colors are in the worms. And so this is something that will happen next year too. We'll collect these and send them to see what the analysis would be. But right now, they seems like the clams are particularly are the ones that accumulate. They're also the ones that build up a lot of fat. And this came out in the Nome Nugget, and this is just another version. So here's Delbert Mugawe here. And this is the clam of the clams, and these, just another view of that map of the stations we did. And those are the locations of the clams, of which they would find these above safety level. And then the other thing I would just point out, you all know this, but all these unusual mortality events. And this system, it's a system, it's systemic, where it's telling you that the system, you know, it's undergoing change, but exactly what's driving the, you know, is it the food? Is it the toxicity? Is it the warm water? It's a combination of those. That's why you need to do it, look at the ecosystem level, the dying of the gray whales, the mortality events, the ice seals, and then the seabirds. And there's no resolution that there's one thing or the other. That is it starvation, but you see a lot of empty stomachs in that, for example, in the seabirds, or is it the toxicity? But you know, when you're sick, you tend to be more susceptible to any type of diseases. So it's, once again, it's a combination of those. And then, so at that point, I think I'd like to move on to let Chelsea talk specifically about the sea ice loss and the walrus diets, because it brings in some collaboration with the local hunters and talking about the changes with the sea ice. And then we can, any other questions, unless you have some right now, we can come back to when she's done. Later. Okay. Good. You're up. Yep.
All right. So I am a PhD student working with Lee and Jackie. And so what I was doing is kind of similar theme as what Jackie was talking about, and is the food changing? But instead I'm focusing on the walrus and that connection to the sea ice, using the sea ice algae as a marker of what's happening with the sea ice declines. So Jackie already showed this figure in hers, but just to reiterate, the way that the system works now really relies on the timing of the sea ice and being in the right place for that spring production, that then there's this really tight relationship with the benthos. And so that ice algae, that early production, sinks to the seafloor and supports these really rich benthic populations, which are then food for the walrus, gray whales, everything we just talked about. And so as the sea ice trends continue to go in the way that they are, or declining, and that we're kind of shifting into this new structure where we have more pelagic grazing going on, we're reducing the quality, quantity, and timing of that food reaching the benthic environment. And so we're kind of displacing some of these food sources for those that depended on the benthos. And so just a quick distinguish between sea ice algae and phytoplankton, as you probably know, but just to clarify. So the algae that I'm speaking about is growing on the underside of the ice. And this is primarily made up of diatoms, a certain type of plankton. And so we also get diatoms representing phytoplankton populations that bloom in the pelagic environment, open water, either as part of the spring bloom or into the summer or fall in some regions. And so because of the conditions of the sea ice where these diatoms can grow, they have a unique nutrient and temperature situation compared to the open water, where it causes the diatoms to produce more lipids. So this early sea ice algae production has a higher lipid content, so could be more nutritious than the pelagic phytoplankton blooms. And so as we talked about, diatoms and other types of phytoplankton can produce these toxic chemicals. And so what I'm going to talk to you about today is not something that's toxic, but it's just another compound that these diatoms produce that are unique to these environments. So, and they can move through the food web similar to the way that the toxins can move through the food web. So what I'm doing is this new technique, it's relatively new, it was developed in the last 10 years or so, but hasn't been applied in this way and in this region. And so, so we have two different types of biomarkers here. So it's biologically produced, it's unique to its source. So when we can identify it in something in the sediment or in an animal tissue, we know that where it came from had to have come from this single source. So you don't need to worry about these names, but this blue one here is IP25 that is only produced in sea ice by a certain species. And then we have this other one here, this other HBI is what we'll call it. That's it's not produced in sea ice, and it's commonly occurring throughout the world's oceans. So that represents a pelagic phytoplankton end member that we can compare it to. And so what I do is I measure whatever samples I'm looking at, I measure for both compounds, and then I look at the proportions of the two. So if we have more sea ice cover or a certain sea ice condition where ice algae is produced, we'll get a higher value for the blue compound. But if it's a more open water environment, we'll see more of this red compound here. And so we have a scale of where it can fall to tell us something about the sea ice conditions that year, or perhaps where within that region they were feeding, if it was an area that had more sea ice than somewhere else. So I was interested in making this connection with the walrus, since they are feeding on the benthos and that connection with the sea ice algae. And so to do this, it's been found in some other studies that the liver is a good organ for looking for these compounds, because it's a lot of lipids are stored in the livers. And so, and another thing that's good about this is that we know that the turnover rates of the liver, so the rate at which new tissue is being produced and replacing old tissue, is about, it's on the order of weeks to about a month or so. So we know when we measure something in the liver that it represents about the last couple of weeks of something about their diet. And so it only requires a tiny bit of tissue to work with. So that's good to not have to use a whole lot. And what's great about these markers that I'm looking at are very stable over time. So as long as it's just been frozen, I can go back and look at older tissues and look at the composition of it in that particular year. So I can go back and rebuild this historical context of the ice algae in their diets. And so these walrus livers, and given that they go back over time, they were collected by hunters in St. Lawrence Island, and also throughout the north. I got some from the North Slope communities as well. So to show you where these are coming from, so the samples I got from the Northern Bering Sea, from St. Lawrence Island, so Gamble, Savunga, and I also had some from Diomede. And these samples were from 2002 to 2016. And these samples were through the University of Alaska's Museum of the North that I was able to work with to get those. And then I wanted to compare, so these would have been harvested in the spring. And then, so I wanted to compare regionally and temporally, so in time, the differences. So looking at those from North Slope communities were harvested in the Northeast Chukchi Sea and more in this late summer, fall period. And so those came from Utqiagvik, Wainwright, Peard Bay, and Point Lay. And most of the walrus samples that came from Point Lay were due to haul-out mortalities, but everything else was also from subsistence hunting activities up there. And the time span from there was 2009 to 2015 I was able to look at. And then I was also able to look at some samples from Round Island in Bristol Bay from 1997, just to get another, an end member here where we're looking at the fall and something that was open water to compare it to. So looking at these compositions by region, so just to orient you here to this graph. So if it was a higher value, that meant there was more ice algae in their diet than phytoplankton. So the boxes you see in red were the samples that were collected from the Chukchi Sea in the summer and the fall. And those in blue are those that were collected from St. Lawrence Island in the spring. And it's probably hard for you to see, but, and so this is going from 1997 to 2016. And so I only had that 1 year from Bristol Bay, if you recall. So from 1997 is this one box down here that's yellow. And so this is kind of goes with what we would think would happen, where we're seeing a stronger sea ice signal in the diets of those that were harvested in the Chukchi Sea than those that are in the Bering Sea. And also the ones in Bristol Bay in the fall are completely open water phytoplankton, so that would make sense. But so it's interesting to see that we have a statistically significant difference between the Chukchi Sea and the Bering Sea walrus harvests. But one thing that stands out, and we do see some variability from year to year, but one thing that does stand out to me is that in 2012 is 1 year when there is no difference between the Chukchi Sea and the Bering Sea walrus. And if you recall that 2012 was a pretty good sea ice year in the Bering Sea, but it was one of the record low sea ice years in the Chukchi Sea. So I think that this information here does suggest that there is that this connection that we're getting from these biomarkers does tell us something about the sea ice conditions that year, and perhaps how their diet was impacted in some way, and perhaps their lipid content of what they were eating was impacted. So then I just to break away and look at the walrus livers from Gamble and Sabunga. So the blue and the pink that we're seeing here is the male versus the female. So the female are the pink here. And so what was interesting to find here was that for all the years that I looked at, the females always had a higher ice signature than the males did. We also see that 2012 and 2014 were particularly high years for females in the region. Yeah, so I guess what's interesting about this is, you know, it shows something about the differences between the males and the females, and it could be that they are foraging in different locations, foraging on different things. It was suggested to me that maybe this had something to do with their reproduction and calving and the female foraging behavior at that time. So there's more that I'll need to look at here, but it definitely could also indicate that the females have a higher reserve of these valuable lipids during this time too for that purpose. Yeah.
Yeah, me 108 month Island explained that the females migrate earlier.
Yes. Well, so these would have been harvested around the same time, and so like I said, the signal only is there, so they're getting that same period of time. So they would have been harvested at the same time, so it wouldn't be that they harvested them earlier, and that's why it has a signature, because then the males should have that too, and they don't.
Did you get any number samples from the winter?
No. So I was limited to what was available in the Museum of the North Archive. So, and keeping them to that spring, I can at least have this comparison, but it would be interesting to know what that looks like.
I've been out to the island in the middle of the winter, and they do, because where they're located, the ice shifts
Yeah.
water for them. The hunters, and they do harvest walrus. So it would be helpful, maybe, to consider getting some inroads, getting samples to help add to this data.
Yeah, I think it could be interesting, but one thing that to keep in mind too is that although you have the sea ice in the winter and they're there, we wouldn't have that production of the sea ice algae at the time. But it might be that, you know, that there is a connection, and I don't know because this just hasn't been measured in any of these samples before, so it's hard to say. So sure, it would be interesting to explore that and round out, because I have these other seasons to look at, but I don't have any winter.
Are you only looking at
Just right now I am, but the idea was to expand it if I could into bearded seals or whales, but that just getting access to the tissues was a limitation. Mm-hmm. Yeah, no, I definitely think it would be interesting to expand this to other marine mammals in the area.
They're more abundant.
Yeah, absolutely. Okay.
So could your change in the pink and the blue levels between the males and the females, saying that it takes a couple weeks for the, if I understand you correctly, for the liver to.
Yeah.
Now, I may not get this right, but I always was told that the males are not feeding as much during the breeding season, so that would be December to March-ish, getting right into close to spring. They're busy with other, they're busy with their man.
Other
Yeah.
activities.
Other activities, but the females are, the ones that are pregnant, which would be something that a hunter would want to get an animal with a calf, right?
Mhm.
So they would be feeding very heavily, right? She's gonna be very hungry because she just gave birth not too long ago. Would that account for some of it?
Yes, and that's actually, so when we were just out in Sabunga and we were having these discussions, that was mentioned to me by at least 2 people, that was a possibility. So yeah, I think that there is something to consider. And, you know, from what my knowledge of the turnover rate of the liver is based on other literature and other studies using different markers. So it is also possible that they do store them a little longer than what I think. But I think it's safe to say, because we see this variability from year to year. It's not accumulating over years. So it must say something at least about the season that they're in, that their feeding was different between males and females, or their assimilation and storage of the lipids was different. And so just to show you the comparison, so these are the North Slope samples. Again, the pink is female and the blue is male, and this light blue was just uncategorized. It was unknown. It wasn't documented. But there's no difference between males and females in the Chukchi Sea. And I don't have all the data that I had associated with the walrus livers. Some did have age data or condition and things like that. So I don't know if the fact that they were pups or juveniles of some sort and versus adult males, I think I don't have that information. So there could be something at play there. But what we know from walrus migration patterns normally is that it's a majority of the females and the younger males that would be going north, while the bulls would stay down in the Bering Sea. But either way, it appears that when they are following the ice north, that they're eating similarly on the same things. So that to me indicates that what we were seeing in the Bering Sea does indicate some difference in behavior versus what is happening here in the Chukchi Sea. And it also helps with that idea of thinking, you know, how long are they storing these compounds, that we will, sorry, just threw me off. So yeah, and just quickly show you that I also am looking at their food sources, so looking at the differences in different clam types and different worms and other things that they might be eating. And so we do see differences in different types of clams and other organisms based on the way that they feed. So those that are subsurface deposit feeders that are really deep down in the mud and getting their food source from there, they were showing that they had the ice signature longer than all the suspension feeding clams and those that fall somewhere in between. So this also lends support to perhaps different things that they're eating in different regions. And it also has a lot to do with the timing, because we know that I've done some experiments to see how long the clams hold on to this signature. And for them, it's about 3 to 4 weeks as well. So we know that maybe those that are deeper in the sediments just have access to the ice algae longer than those that are feeding from the water column. So this is just another part of the study to try and figure out what's happening there. So just quickly in summary, I'm trying to figure out more about what this difference between the males and the females means, but I do think that what we can take from this is that as the sea ice continues to retreat and retreat earlier and further north, that it might have more of an impact on the females than it does the males. So remains to be seen there, and that there might be different feeding preferences. And what I'm also interested to do is look to see if there's any relationship between these ice signatures and the condition of the walrus. So I don't have that data that was received with the tissues, but I can run some other analysis on this, looking at the total lipid content and also the stable isotopes of these livers, to see if there's something I can say about the connection of where they were feeding in the food web and what the overall condition might have been. So that's something else I'm looking into. And then also just taking into consideration that humans some of you all are a part of this food web as well, and that these changes to sea ice and these shifts to the food web also impact you and the quality of the food items that you're receiving too. So that's not lost upon me that we're focusing on the walrus, but this also impacts you. And again, these differences in the levels that we see, they don't necessarily mean that it's a bad thing, but it does indicate a symptom of greater shifts that are happening. So, yeah, with that, I think that's where I'll leave it. And we'll thank you all for coming out. And does anybody have any questions? Yes.
Taking liver samples, how old are you? Does the liver have to be a quality for your research?
So long as it's just been frozen and not in any sort of chemicals, it's good to go. So as old as there is.
Okay, so I go out hunting and get a walrus. I come back to know. Just a few hours or a day, getting one,
Yeah.
Yeah. So the question I was asking, in that
Okay.
timeline, we're not necessarily being able to put it in freeze.
Right. Yeah, so I don't think that has any impact on these particular compounds that I'm looking at. They don't degrade that quickly.
I noticed you don't have any from our region.
Where,
Sure.
oh, okay. Well, I honestly, I took what was available from the Museum of the North.
Permitting issues.
Yeah, and this was just, you know, a pilot study. I didn't have any other pre-existing data to work from, so I wanted to just, what's available, I can look at that and then go from there. So I started with those with the Museum of the North. I saw some interesting results. Then I reached out to the North Slope Borough Wildlife Management with Rafaela Simmelmeyer and was able to get some more there. So I'm kind of building on this story as I can get my hands on samples.
Building them.
Tissues are donated. You don't need a permit.
Well, no,
Well, there's still a process to go through. And I needed to be on the permit for North Slope samples.
Right.
But by working through Museum of the North, they have a permit overhead that I fell under. So I couldn't just, you know, come to your house and get a walrus liver from you without getting,
Huh?
yeah.
It becomes no longer a subsistence, it's no longer a subsistence item.
Right.
You should especially, know that, keep that in mind.
And like she has to keep a record of how much tissue she used to report back to the, like Raphael, where she can report back on her permit that, you know.
There's a paper trail of
There's a paper
what I've
trail.
used. And
We don't use all the tissues, we're looking at either
and the Museum of the North had a very rigorous process of, I had to tell them the absolute maximum amount I needed. And so they would only give me a teeny tiny bit to preserve for other researchers for other purposes. But with the North Slope samples, they were able to give me a little more, and then I'll take what I need and then give it back.